Today we will be discussing Dreifuss muscular dystrophy or EDMD, a condition that primarily affects muscles used for the movement and heart. It is characterized by muscle weakness and wasting that slowly worsens over time, beginning in muscles of the upper arms and lower legs, and later also affecting muscles in the shoulders and hips. Many patients also develop heart problems by adulthood. Among the earliest features are joint deformities called contractures, which restrict the movement of certain joints, most often the elbows, ankles, and neck. These usually become noticeable in early childhood. EMD is caused by mutations in several genes, including EMD, FHL1, and LMNA. Today we will be focusing on EMD. More specifically, we will be looking at the X-linked form, meaning that the gene causing the trait or disorder is located on the X chromosome. It has an estimated incidence of 13 in 10 million. EMD usually makes a protein called emerin, which is a pointed-end, F-acting capping protein located in the inner nuclear membrane. We will get back to that later. Most EDMD patients harbor nonsense mutations and have no detectable emerin protein. There are 3 EDMD causing emerin mutants: S54F, Q133H, and the changed amino acids 95 to 99 that localize correctly to the nuclear envelope. Mutation can also prevent the protein from being made at all. Emerin interacts with other proteins as well as binds to lamins. Lamins, in case you didn't know, line the inside of the nuclear membrane, where they provide a platform for the binding of proteins and chromatin and confer mechanical stability. As mentioned previously, emerin is a component of the inner nuclear membrane. It contributes to numerous cellular processes, including chromatin tethering, gene regulation, mitosis, nuclear assembly, and development signaling. The nuclear membrane is composed of a double membrane structure that surrounds the nucleus, acting as a barrier between the nucleus and its DNA and the surrounding fluid, aka cytoplasm inside the cell. Additionally, it is permeable only to small nonpolar molecules and has pores that allow for the regulation of the movement of molecules such as proteins and RNAs in and out of the nucleus. The nuclear membrane's main function is to act as a barrier that separates the contents of the nucleus from the cytoplasm. It is continuous with the rough endoplasmic reticulum, or organelle, important in making and transporting proteins. Both the rough endoplasmic reticulum and the outer nuclear membrane are covered in ribosomes. The mobility and expression levels of the mutant forms of emerin are indistinguishable from those of wild-type emerin. However, mutant forms of emerin have weakened interactions with nuclear lamina components. This then weakens the structure of the nuclear envelope in cells that undergo a lot of mechanical stress, such as skeletal and cardiac muscle cells, making these cells more fragile. Both of these combined mean that ruptures in the nuclear membrane are more frequent. When a nuclear membrane ruptures, it leads to intermixing of nuclear and cytoplasmic contents. Ultimately, these mutations result in muscular dystrophy. No specific treatment exists for EDMD. Treatment is aimed at the specific symptoms present in each individual. It may include physical therapy and active and passive exercise to build muscle strength and prevent contractures. According to Reddit user Genie_cat333, 2 common supplements taken are coenzyme Q10 and creatine. Exogenous incorporation of coenzyme Q10 into the mitochondria can double the oxidative activity of NADH, thus providing metabolic support to muscle. Creatine is a hydrophobic molecule bound to the inner mitochondrial membrane that functions as an electron acceptor for complexes 1 and 2 of the respiratory chain. Creatine has a role in high-energy phosphoryl group transfer during skeletal muscle contraction via the creatine kinase reaction. It also plays a role in transferring energy from the mitochondria to the cytosol in tissues with a high energy requirement, such as brain and skeletal muscle. Additionally, creatine may play a role in the regulation of muscle protein metabolism. Because EMD mutations are almost always associated with a lack of emerin expression, this makes X-linked EDMD theoretically amenable to emerin replacement gene therapy.